I would like to welcome everybody. My name is Professor Carola Dillenberger. I'm the Director for the Center for Behavior Analysis at Queen's University. Hi, everybody, and welcome to the Center webinar series of 2025. I'm absolutely delighted today to introduce you to AlicjaPus. I don't know if I pronounce this right. You can keep us right.
Oh, that's close enough.
It's close enough. I met Alicia in Poland a few weeks back, and I heard her speak to us at that meeting, and I just could not wait to have her here and welcome her at the Center for Behavior Analysis to share her vast amount of knowledge as a neuroscientist, science communicator, social activist, and also behavior analyst. So I'm delighted to welcome her here today. Alicia is affiliated to the Nenki Institute of the Polish Academy of Sciences. Her research explores the integrated relationship between the brain and behavior, with a focus on motivation, emotions, and social influences on decision-making. Her interdisciplinary approach combines life sciences, behavior analysis, and advanced computational methods. During her graduate studies, she designed and co-patented the EcoHub, a system for naturalistic assessment of social behavior in laboratory animals, now widely used across Europe and North America. Throughout her career, she has been a strong advocate for equality and mentoring. She co-founded and led numerous organizations, including her PhD Students' Council of the Polish Academy of Sciences, the Professional Development Committee of the Yale Postdoctoral Association, and Women in Science at both Yale and the Nenki Institute. For many years, she also served as a biology educator for Khan Academy Poland. Currently, she is a member of the Global Diversity Committee of the albeit Network and an ambassador for the Polish-US Fulbright Commission. It is an absolute delight to introduce or to welcome you to the Center today, or it's the evening here in Northern Ireland. And to know that we have a very large audience from all over the world, we will have a look at the different welcoming messages from everybody, but generally speaking, they will be from everywhere. Alicia, I will hand over to you. I will switch off my video feed so that people can just concentrate on your presentation.
Before you do that, I just want to warmly thank you for this invitation. It means a lot to me to be able to speak on a platform that is dedicated to behavior analysts, because that's the discipline that is so close to my heart. Thank you kindly. And I am delighted to give this lecture, so let's give it a go.
Okay, let's go.
And at this point, I would like to thank you all, the audience that is tuning in, for spending this very, very interesting, I hope, very interesting night with me. Today, I would like to invite you to think about the brain as a behavior generating machine. So we are going to be focusing on neurodevelopmental disorder, because that's the most important angle that I would like to converse with you today. However, before we get there, I just want to tell you that I divided this lecture into 3 parts. First concerns how the brain works. What is the current view of neuroscience on this behavior-producing machine? The second part is going to be about the most cutting-edge methods in neuroscience that at this day and age allow us to manipulate behavior in real time, because I would like you to know how far we have come from the black box analogy. And the last part is going to be about putting it all together to better understanding of neurodevelopmental disorders. So as I said before, brain is the place where everything in our behavior starts. But it's not only our behavior, it's also the broader definition of behavior, which concerns thoughts, feelings, emotions, you name it. For the purposes of our today's presentations, I am going to be talking a lot about the brain function. In fact, if I was to choose one word for this entire lecture that we would be focusing on, or one phrase rather, it would be the functional neural circuits. And in a minute, you will be able to tell what they are. So let's take a look at how the brain works. This is a very tiny organ in our head. It weighs less than 1 kilogram even. And when you think about it, at its very core is the generating of everything we do in our life, most notably the behaviors. They are encoded in the patterns of neuronal activity, those flashing lights that you see now on the screen. This is the activity of the neurons that are composing our brain. And in the sequences of those signals, which are normal electrical signals, is encoded everything we do, everything we think, everything we feel. From the most mundane behaviors like brushing our teeth to our dreams and our plans and goals for the future. So taking that perspective really enables us to understand something that wasn't so clear 20, 30 years ago. Brain is not operating as a combination of many structures. Probably you all have seen pictures like this one before, just assigning the particular lobes of the brain cortex. Of course, it is like this, that certain brain structures or areas, as they are oftentimes called, they are very important for specific functions. For example, the visual cortex is important for vision. But it's not only the visual cortex that makes us see. It's many different neurons in many different brain structures. So from this brain view, we have come to what I am showing you on the screen right now. This brain on the left-hand side is the brain of a mouse. We'll be talking about mouse quite a bit today. And with those different colors, you see the neuronal connections throughout the brain that are composed of different neurons that are connecting and talking to one another. So today, I would like to encourage you to think about the brain not as a set of structures, but rather about something that is the collection of neural circuits that are connected throughout many different places throughout the entire brain. And here comes the word of our lecture today: functional neural circuits. So the function, the behavior, the physiological response, the thought, you name it, comes from the activation of the set of cells that are located in many different interplaying places within the brain, as we can see on the left-hand side. And those neurons activated together in a particular context, particular time, particular place, they make this function to be presented. For example, I can grab with my hand something that is within my reach because a particular circuit that is responsible for hand grabbing is activated within the brain. A very important thing to understand about the brain is that the cells that are composing it, neurons, are a very special type of cell. What is rarely talked about is that neurons are extremely rich in energy production and energy usage. It is because they need a lot of energy to send all of those signals that we have been talking about so much. But beyond being very energy hungry, they have many different characteristics that are differentiating them from other cells in our bodies. First and foremost, we should always remember that neurons really are not being produced on a large scale, at least throughout our lives. What we are born with actually gets reduced very severely within first months and years of our life. And what we are then stuck with sticks with us till the end of our life. The other thing that is very characteristic is what we are seeing on the screen right now. I am not sure you can see my pointer, but we see the neural networks that can be formed, the networks that I have been talking about previously. They only can be formed because all the neurons carry on their surfaces the connections that we call synapses. I am sure that you've heard of them, but let's stop for a second and try to understand synapses a little bit better. So on this right-hand side picture, you can see a neuronal cell the blue stuff is the cell body. And as you can see, there is many branches that are coming out of the cell body. Those are called dendrites. Some of them are called axons. Those are the branches that are sending the signals to other neurons or receiving information from them. What is very notable here is the teeny tiny yellow dots on those branches. Those are synapses. And do you know how many synapses are there on the average cortical neuron in the human brain? There can be as much as 10,000 different synapses on them. Can you imagine knowing 10,000 other people or being connected to 10,000 other entities that can actively send signals to you? All this information that is coming from different neurons is integrated in the cell body, processed there, and based on this information, the decision is made on whether or not this particular neuron itself, the one that we can see in the picture, should send the signal to the neurons that it itself is connected to. So what kind of complicated system this can create? And when you take under consideration that there is billions of neurons in our brains, this really makes you wonder and understand what kind of complex activation patterns and in result behavioral patterns can be produced out of that. Beyond having synapses, the neurons are very characteristic because there is not one type of neuron in a brain. In fact, there is many different types. What we are seeing on the screen right now is a picture representing hippocampus. This is one of the brain areas that we know is very critical for encoding context, the particular time, place, and the circumstances that are surrounding a particular behavior, or for the formation of memories about where, when, what we call episodic memory. Hippocampus is very important for that. And in the beautiful project in neuroscience that was called Brainbow, the scientists took an aim at understanding the diversity of neurons in this brain area. And different colors that you are seeing in this picture, they are representing different cell types. And this is only scratching the surface of how diverse they really are. There are neurons in the brain that are excitatory, meaning that they are sending signals to other cells that are making them more excited, more probable for them to send the signal further. We have neurons that are inhibitory. But within those two broader groups, we have huge diversity among different brain areas. But even as you can see, within a certain brain area, one next to the other, you can have neurons that are very diverse. So all this collage of information is created out of diversity of neurons and their function, out of the diversity of the connections throughout the brain, and out of the diversity of the synapses that really thousands of them are located on one single neural cell. Taking all of this under consideration really makes us wonder, what are the origins of the function within a brain? We already know that this has to do with particular activation patterns among those diverse neurons. But when we are looking at a more macro scale, it turns out that the neural underpinnings of mammalian behavior are really similar in many different species. What you're looking at right now in the screen is the comparison between the brain of the monkey, of the human, and of a rodent. The rodent is here, as I've mentioned before, not by accident. Mice are, in fact, the most commonly studied species in neuroscience these days. So we'll get back to this. Thought in not so long. But let's take a look at the analogies between the brain of a human, the brain of a monkey, and the brain of a rodent. What you see here is the sagittal cut of the brain, which means that we would cut from front to back, and you can see the inside like when you would open an apple in two parts. And the areas that are here in blue are the analogous areas between those three species. In other words, even though human brain is way more complicated than the brain of a monkey or the brain of a mouse, the critical elements that are there in every one of those species. For example, the areas that are responsible for our ability to experience rewards, such as nucleus accumbens, is in the brain of a human, in a brain of a monkey, and in a brain of a mouse. Same when it comes to the areas that are responsible for aversive stimulation. Same when it comes to the areas that are responsible for so-called higher function and social behavior. So we can say, after Charles Darwin, and this is one of my favorite citations from him, that the difference in mind between men and the higher animals, great as it is, certainly is one of degree, not of kind. So again, the takeaway from this is that, in fact, yes, the brain of a human is way more complicated than the brain of a mouse, but still, the critical elements of it, and even more importantly, the biophysical processes that are making this brain work are very, very similar in both those species. So it doesn't really matter from the point of view of a behavior analyst if hello is said like this, or like this, or like this. What is important is the function of this behavior rather than its motor component. And for this very reason, we can find the analogous neural circuits, let's stick with social behavior, that are responsible for getting acquainted with new individuals in a brain of a human, in a brain of a mouse, and in a brain of a dog. They are not going to be identical, but the motivation for this specific behavior is going to look very similarly in the neural circuits of all three species. So as I said before, mice are very important in neuroscientific research nowadays. When it comes to mammals, there is no other mammal that has been studied so extensively by this discipline of science than mice, for two reasons. First and foremost, they have very similar brains, even though simpler than humans. And they also have a very rich behavioral repertoire. In fact, they form social networks, they compete, they learn socially, they are very flexible in the ways they can behave in different contexts. They form social groups, can change them. So this is a very important characteristic of this species. You can test a lot of interesting behavior in it. And also there is one more thing. We have a lot of these days transgenic strains of mice that allow to pinpoint the specific types of neurons that I have talked about, or dysfunctions of neurons, even more importantly, and study their function within the animal. The most common thing to do, and we will be talking about an example of this today, is to take the mutation or the gene dysfunction that is found in clinic, because, for example, it has been identified as a potential cause of, let's say, schizophrenia. Mimic this genetic impairment in mice and study such strain of mice as a model of the disease that has been identified in a clinic. So having said that, there is another side of the coin when we were talking about the fact that the behaviors can look in a different way, but the most important thing is their function. The other side of the coin is that we have behaviors that look exactly alike and they have very different functions. So here is the example of somebody writing hateful comments online, and this is the kind of a social behavior. But we have another person who looks exactly the same to the person who's looking at them on the side, and they are typing an essay, let's say. So from this point of view, oftentimes it's really hard, without looking into the brain, to understand the animal behavior in detail when the context is very similar or sometimes undistinguishable to the eye of the observer, but the motivation for doing the behavior is very, very different. And looking into the brain actually allows you to figure all that out. At least sometimes it does, if the experimental design permits it. So coming back to the functional neural circuits, putting it all together, we have, let's say for affiliative behavior, a set of brain structures, of brain areas that are involved, specifically very concrete neural cells within those areas that are interconnected and are playing a role in this function, can be called dysfunctional neural circuit. But within the very same brain structures, and again, we are seeing a sagittal view of the brain, which means we cut it in half. This time it's the brain of the rat, but we know at this point that it doesn't change much. We can see that when it comes to competitive behavior, which is completely different type of social behavior, very similar brain areas are going to be involved, but the functional neural circuits within them are going to be different. It can be the same cells, can be different cells, they can be connected differently, they can lay one next to the other. So just by this, I am trying to tell you that it's not enough to look at the specific brain structures to understand behavior. You have to look within them to really understand what is going on. And this is a very good example because we have two polar opposite types of social behaviors. One is affiliative and the other one is based on aggression. And we have very similar brain areas that are involved, but the neural circuits within them are very, very different. So having said that, let's take a look at the latest toolbox of neuroscience. What are the cutting edge techniques that really allow us to study all of the complexities that I have told you about? Not only to look at the particular brain structures, but look within to see what happens when the animal is really behaving. And I divided this latest toolbox of neuroscience subchapter of our lecture today into two parts. First of them concerns what we can see in the brain. I would like to show you how cutting edge it is and what granularity we can really afford these days. So first and foremost, these days it's feasible to look into the brain of a behaving animal in real time. The most commonly used type of method for this is so-called two-photon microscopy. This two-photon microscopy method allows us to see different types of neurons. In those square-shaped images, what you see are different types of neuronal cells within the same brain structure. They are of the same color because the two-photon microscopy only allows you to register how bright the cells are, but nevertheless, this is amazing because in different pieces of the same observation, you can look at very different populations of cells. And again, I am not sure if you can see my cursor, but what I am pointing to right now is that you can even observe the neurites, meaning the dendrites and the axons of the cells in the behaving animals. And if you have a good enough microscope, you can even take a look at the synapses. So I would like to show you a brief movie. Oh, you can see the cursor. That's amazing. Thank you. So this brief movie is showing you what we previously saw on the animation in the human brain. You can see the real life flashing neurons of a mouse, which is behaving under the objective of the two-photon microscope. Those flashes mean that the neurons got activated. This is really an amazing technique, which allows you to see not a very broad picture that EEG gives you or the MRI, which you only are able to go as far as see specific areas or specific substructures. You can really see the particular neural cells. And this amazing method allows you to do something that was really not feasible not so long ago. It allows you to align the activity of the specific neural cells within a brain with specific behaviors. What I am showing you right now is real experiment movie from the visual cortex of an animal that is undergoing visual learning. So this animal is set a task of differentiating between two different types of stimuli. So what you can see marked with those circles are 3 different neurons. And in real time, you can see the activity of those neurons being drawn on a screen. You can see when the neurons are activated. And this can be aligned with the behavior of the animal. And you can figure out which one of them is actually involved in producing the behavior that you are observing. Because obviously, the activity of the brain, which is the source of behavior, comes a little tiny bit of time before the behavior itself appears. From this perspective of the 2-photon microscopy, as I said, if you have good enough equipment, you can also see not only the flashing neurons, but also the synapses. And here is something that we call a passive scan of the synapses and dendrites in the brain. The red parts are the dendrites, and the flashy yellow-green things are the synapses, the same that we saw before in the static picture in one of the slides. I am going to play it one more time for you. So we can look into the brain in real time. We can take a look not only at the activity of the neurons, but we can take a look at something that we call synaptic turnover, which is how synapses change as a function of learning as the behavior progresses. So this is it. When it comes to observing the brain, what is at the forefront is actually what we saw. But there is another part that is even more exciting from the point of view of the behavior analyst, and this is the regulation of the behavior. We have numerous methods right now that enable us to change the activity of the neurons in real time. However, some of them went out of fashion. What was very popular in the 20th century up to, let's say, the 80s were the lesions and electrical stimulation. The simplest experiments with brain manipulation went like this. We put the electrode in a brain, drive a current through it, and see how the brain responds. Because the brain works with electricity, it was indeed very easy to change what the brain does with this method. Another method was taking away parts of the brain, lesioning them, getting rid of them, and seeing what kind of functions are being impaired. But this day and age, we have way more sophisticated methods. First of them is pharmacology. And by this, we can obviously say when we are taking pharmacological substances, any kind of psychoactive drugs, for example, this is what we are talking about. You're taking something and this changes the activity or the function of the neurons in your brain. But in current neuroscience, this kind of technique can be much more intricate because we have what we call cannulas. Those are teeny tiny glass needles that you can put in the particular place within the brain, release the substance exactly in this tiny spot, and see what it does to the activity of the small group of neurons of your interest. Another method is genetic manipulation, and this is something that I have referred to before when I told you that it's very common that the new strains of animals, I was talking about mice in that instance, are being created in response to our knowledge about specific genetic deficits that are driving neurodevelopmental or neuropsychiatric disorders. But by far, the two most common methods that are used to regulate the brain function today are optogenetics and chemogenetics, those two. And I would like to focus on them on a little bit more because they are very prevalent. And in my opinion, they are the future of behavioral regulation via the means of neuroscience. Optogenetics is regulating neuronal activity via the means of flashing light. Chemogenetics is regulating neuronal activity by supplementing the organism with otherwise non-functional, in a physiological way, substance. So let's take a look at those methods. They are both lock and key methods, meaning that you need two components for them to work. You choose a particular type of a neuron that you are interested in. Or if you are interested in all the neurons, you can do that too. But the more intricate experiments usually involve choosing the population of neurons of your interest, because you already know that different neurons have different functions, even within the same functional neural circuits. And then you take an animal, can be mouse, can be fly, can be any mammal. It actually works in humans. There are clinical trials with the use of optogenetics. We'll be talking about that in a minute. So what you do is you put the receiver in the neurons in the chosen area of the brain. You put there the ion channels that are responsive to particular wavelength when we are talking about the optogenetics, or to the particular chemogenetic substance when we are talking about the chemogenetics. I would like to stress the fact that the very substances that are used in chemogenetics, they are inactive in any other way. So if you took them, or if animal took them without being prepared in a way that I am talking about right now, they would just go through the organism and nothing would have happened. You need this lock and key rule for them to work. So when you have the receiver in the brain, meaning that you have made the neurons susceptible to the particular factor, you can activate the neurons either with the use of light or by the delivery by injection, most commonly, of the substance that is going to activate the neurons. The key difference between chemogenetics and optogenetics is that optogenetics is very precise. You can cause the changes in neuronal activity here and now like this. It's with millisecond precision. Chemogenetics, not so much, because you deliver the substance to the organism and it is staying there for an extended period of time. But what is critical is that from my particular scientific interests point of view, chemogenetics allows you to manipulate behavior for extended periods of time. And I am going to give you an example of that. So again, optogenetics is very precise. You can change the behavior here and now, but you cannot use it for very long. Let's say you would not be able to manipulate behavior for longer than an hour. In chemogenetics, you can manipulate behavior for extended time periods, but with less temporal precision. So having told you about how it works, how about we see some movies that are going to show us how it looks like in practice? So in this first movie, let me stop it for a second. In this first movie, we are going to see a mouse which has been prepared in a way that I have just described. So it has undergone the delivery of the sensor to its brains. And right now on its head, it has a wire that is delivering the light when the experimenters decide for the light to be delivered. And every time the light is delivered, the activity of the chosen neurons are manipulated. Let's take a look at what kind of power this technique may have. We have a mouse that is presented with a wooden stick. When the laser is off, no light, it doesn't interest the mouse. But when the laser is going to be on, we are going to see a very different type of behavior. In this case, immediate attack. Because the neurons that were manipulated in this experiment are responsible for aggressive behavior. When the laser is going to go off, the aggressive behavior goes away. And let's take a look at another object to see that that's not specific for the stick. When the light was off, it was of no interest to the animal. But when the light went on, it immediately attacked the object. There is no threat to it. There is no reason for this animal to behave this way in this particular context. So again, the activation of the particular neurons, which researchers knew were responsible for eliciting aggression, allowed to cause this behavior on demand. And here's another type of behavior, this time reward motivated behavior. What is important to understand in this example is that this animal has not been food deprived. It has eaten a lot prior to this experimental setup, and it has no reason to eat out of its own volition. So what we see here, those white elements, this is food, very attractive food, but not attractive enough to cause consumption in and of its own. And again, we have an animal that is correctly prepared, and we will see the laser going on and going off, and let's observe the behavior of this animal together. When the laser was off, food was not interesting, but this animal starts consuming as soon as the light goes on. And it will continue to consume as long as the light is on. Now the light is off, and the animal lost interest in food and goes to explore the rest of the experimental environment. Again, the light will go on. It catches the first available piece of food, and it starts consuming right away. So you can see that optogenetics, as I told you before, when you understand the neural circuits that are involved in a given behavior, allows you to control this behavior with huge precision, huge temporal precision. But the downside of this method, again, is that you cannot do it for a very long time. So those were two examples that required a wire. I just wanted to show you now that at the forefront of neuroscience, the wireless methods with the lights are more and more popular. This is no causative experiment. This is just to show that you can use wireless optogenetics without a wire in the groups of animals and thus manipulate their social behavior in this way. And finally, and most importantly, there are already different types of trials of using optogenetics in humans. So here you see a person that has retinopathy, which causes them to be blind from the moment that they were born. This is a very example of a genetic disease that can be manipulated with optogenetics. So what did researchers do? They injected those transmitters that we were talking about into the eye of the person that is here on the screen. And the goggles that the person is going to put on, they emit the kind of light that can artificially activate the neurons that have been manipulated to be activated for the very first time. And this video shows this person being able to see the elements of shapes for the very, very first time in their life. It's in French, but there is subtitles. So what you see is the person that is without the goggles that would deliver the optogenetics. And they cannot see very well. They cannot see anything. It's like always. But when they will put the goggles on, which happened right now, they will be able to say whether or not the object is on the table, and they will be able to say where it is located. And they are going to reach for it with their hand. There we go. That's another trial without the goggles, so without the workings of the optogenetics. It's a smaller object, and now with the goggles, and again, the person is able to see the outline of the shape on the left-hand side, and is going to, again, be able to reach for it. This time, I believe, with the left hand. Oh no, the right hand. They are trying. There they go. So as you can see, this is not just pie in the sky ideas that I am talking about. This is something that is really happening right now in the future of the medicine and the future of neuroscience. So when it comes to testing the behavior, there's also a lot of methods in the lab that are enabling to test very complex behaviors. One of such systems was invented in our laboratory a number of years ago. And the most important element of it is that you can test a lot of mice at the same time in the environment which they are leaving. So this enables us to do very interesting experiments such as this on understanding how the brain is involved in meshing together two unfamiliar social groups. In this experiment, we are dividing the territory in the experimental environment into two, and we are keeping animals separately. And then on one day, we are removing the perforated partitions that are here, and then they can be together. And we are studying what is happening at the beginning of those encounters. Can we facilitate, or on the other hand, maybe we can do something bad to the behavior of acquainting the new social groups. And I am introducing this setup to get you acquainted with the results that were obtained with the chemogenetics method, the one that I told you that is not as precise as optogenetics, but it enables us to manipulate the longitudinal behavioral patterns, such as in this experiment, when we want to observe and manipulate the behavior for extended period of time, many hours. So those two graphs, they are showing affiliative behavior on the left-hand side, and on the right-hand side, we see the competitive behavior. Those bars that are visible right now are for non-manipulated animals. This is for the in-groups, animals that knew one another, and the black one is for the out-groups, animals that have not acquainted one another. But what is intriguing is that when we manipulate the brain with chemogenetics in a specific way, we can completely diminish the affiliative behaviors. And on the other hand, we can crank up the competitive behaviors, which even more importantly seems to be, this is a pilot experiment, so it's too early to say, but it's most likely characteristic only for the animals that have not been acquainted with one another before, which is even more intriguing. So as you can see, not only you can cause the behavior here and now to change, but you can also, with the means of chemogenetic, influence longitudinal behavioral patterns. So it's time to better understand the neurodevelopmental disorders through the lens that I have just shown you. At this point, you are able to understand what is at the forefront of neuroscience, what are the functional neural circuits, what methods are used to observe them and manipulate them, and thus manipulate the behavior. Let's take a look at neurodevelopmental disorders from this point of view. Because the brain is the source of function, it is also the source of the behavioral dysfunction. And the contemporary psychiatry and contemporary neuroscience really tries to see the neurodevelopmental disorders through this lens as something that is being caused by the dysfunctions of the nervous system. And let's focus on autism as one of the most prevalent and also most troublesome neurodevelopmental disorders, and try to understand it from the biological point of view. First and foremost, autism is very highly hereditary, which means that you have a higher chance of being autistic yourself if somebody in your family has autism already. So this means that the genetic factors are very, very important, but monogenetic causes of autism, meaning the causes when there is one gene being impaired, and this causes the autism to happen, this is very rare. There are monogenetic causes. Fragile X syndrome gene impairments are one of those causes. We are going to be talking about an example of that today. But most of the cases are so-called polygenic, which means that multiple different types of genes are being impaired. And of course, the environmental factors are very important. There are several prenatal factors of biological kind that have also been identified as increasing the probability of occurrence of autism. But you already probably figured out that we are studying those biological causes to be able to propose a very much personalized therapy that is going to talk to the particular nervous system elements that are impaired in a given patient. Because what is most important is that genes that have been found to be responsible for the occurrence of autism are the ones that lay the proper function of the central nervous system. Most of those genes are being expressed in a developing brain. What does it mean? When you have a gene, let's say it's a gene that is involved in a nervous system, it doesn't mean that it produces proteins at the same level throughout your life. Some of the genes are most important, the production of the proteins that is created by them is most important when the brain grows and develops and matures. So a lot of those genes that are important for the development of the brain their impairment specifically, have been identified to be associated with autism. And very many of those genes are influencing the synaptic function. And we will talk about that in a minute even more. Many of those genes fundamentally alter early development and are linked to more than one neuropsychiatric or neurodevelopmental disorder. So, for example, many genes that have been identified to be involved in autism, they have also been identified in depression, schizophrenia, and so on. So malfunction of the central nervous system of a particular kind can be associated with many different disorders. In autism specifically, the function of the brain that has been reported to be impaired is so-called neuronal plasticity. Neuronal plasticity, both throughout the development of the brain and in adult life, is the ability of the brain to change itself upon the demands of the environment. And here comes learning. So neural mechanism of flexibility when it comes to behavior, emotion, motivation, they are all based on this ability, physical ability of the brain to reshape itself, change not only the patterns of activity, but most importantly, the connections between many different neurons throughout the functional neural circuits. This ability of the brain, neuronal plasticity, can be too high or too low, and both cases are not really great. When you are too flexible, you cannot learn and establish any stable behavioral patterns. When you are too rigid, well, we know what happens oftentimes with autistic individuals who have trouble with being flexibly able to adjust their behavior when changes in the environment happen. So neuroplasticity is a very complex phenomenon on many different levels. First and foremost, it can be the strength and flexibility of the synapses that needs being changed. So we have a synapse, it already exists. We can make this connection already existing between the neurons stronger, or we can make it weaker. Another thing is that you can change the number of synapses. Some synapses can just disappear. And as we saw on this dynamic scan in the brain, we have synapses that can appear and we have synapses that can disappear throughout the behavioral changes. Also, neuronal excitability is something that can change. So we have a neuron, and this neuron can be responding weaker or stronger, depending on what it, quote unquote, learned in interactions with the environment. And finally, this is not a very important from the point of view of the whole brain phenomenon, but it exists nonetheless. The number of nervous cells can differ. So there are specific regions of the brain, one of them being hippocampus that I have previously mentioned, where new neurons can appear throughout life, but they are not very numerous. We still don't fully understand what new neurons do and what kind of functions that they can gain as they are being born in adulthood. Nevertheless, it's a type of neuronal plasticity that we have yet to understand. I have previously mentioned the synapses being the most important focus in autism because essential molecular machinery that is keeping the synapses together. And here we see a synapse at work. We can see the signal being sent, and let's play it again. We can see the signal being sent when the synaptic vesicles are being released between two different neurons, and we have this flashing light sending of information. This essential machinery that make them work is indeed very intricate. And a lot of the genes that are responsible for those synaptic proteins have been identified to be impaired in neurodevelopmental disorders, specifically autism. Those dysfunctions of synapse are called synaptopathies. The dysfunctions of synapse that are laying the foundation for neurodevelopmental disorders. Malfunctioning synapses, the causes of the malfunctioning synapses are called synaptopathies. And this is a key cause of autism from the biological point of view. What you see right now is the synapse from a slightly different perspective. This time, those different colorful elements here are different proteins that makes this synapse work. On the side where the signal is being sent and on the side where the signal is being received. You can see, and those all, by the way, have been identified that when they are impaired, the synaptic plasticity goes away, and all of the proteins that you see here have been in some way associated with appearance of autism. So you may see and understand how intricate machinery it is. What is most important to understand about it is that there is many different functions that need to happen for the synapse to work correctly. It has to be held together by the complexes of proteins that are neurexins and neuroleukins, such as here. The signal has to be sent, it has to be received. Some of the proteins, one important one that we can see here, FMRP, are responsible for the production and delivery of other proteins to the synapse for it to work correctly. So you may see that a lot can go wrong. And this is maybe one of the most important elements that I would like you to remember from this lecture. A synapse needs a lot of different elements for it to work correctly. And even if one of them is not working correctly, there is a high probability that synaptic plasticity will not be happening as it should, and it may cause behavioral impairments. The protein that I have mentioned before, FMRP, is going to be an example of the targeted therapy that I would like to tell you about, and this is why I marked it here, so that you saw that it's a key player in this machinery that we are talking about. So taking it all together, the very modern approach to the treatment, biological treatment of neurodevelopmental disorders is first understanding that because the functional neural circuits underlying different behaviors are different, the different symptoms, even within the same neurodevelopmental disorder, are going to be caused by different mechanisms in the brain. So for that very reason, that you may have different mutations and dysfunctions of the gene causing problems, different neural circuit causing problems, the tailor-made approach is something that the scientists are currently striving for. So what would this targeted approach to the treatment of neurodevelopmental disorder entail? It entails three things. The intervention needs to be precise and targeted at the well-understood biological mechanism that is underlying a well-defined clinical problem. In fact, there are three levels of specificity that need to be addressed. First of them, we need to understand a particular symptom. In other words, the behavior that is deficient that we are looking at. It's not occurring as it should. And we are interested in this specific behavior, not all-encompassing phenotype of a given neurodevelopmental disorder. Secondly, we have to well understand and have defined the neural circuit, the functional neural circuit that is involved in this behavior, that produces this behavior. And finally, we have to understand what is wrong within the circuit. What kind of molecular malfunctions are causing this circuit not to work as it should? There are different levels on which we can target the molecular malfunctions. within the neural circuits. Most of the targeted approaches are targeted at the DNA level, some of them at the RNA level, and the most challenging is the protein. Proteins are what really makes the neurons do what we want them to do. You can think about them as the building blocks of the function that is realized by each neuronal cell. So this level is at the same time the hardest, but the most attractive from the point of view of proposing new therapeutic interventions. This is just to show you that there are ongoing clinical trials for different types of targeted approaches to neurodevelopmental disorders. This is a table, so it's not very visible, but I am going to tell you what you can see. Here is DNA level and the RNA level, and at what is the current status. Some of them have already been approved in the United States, the targeted approaches. And unfortunately, none of the approaches that have been tried so far when it comes to the protein level has been efficient in autism. But I would like to tell you that we came up with an experimental paradigm, I'm going to show you in a second, that may be effective in this manner. So what I'm trying to say with this slide is that a lot of people are trying. Some of them have been successful. There is still miles to go, but we are definitely making significant strides in this direction. So the experimental therapy that I would like to tell you about that we have developed was tested on the animal model and met the criteria that I have introduced before. We have a well understood model in which we know what is wrong. Those mice, fMRI1 knockout mice, they are a model of fragile X mental retardation gene 1. They have no functional copies of this gene, so it doesn't work. This is modeled after the patients because fragile X mental retardation gene 1 has been identified as a monogenetic cause of autism. In fact, the most frequent monogenetic cause of autism in the world. Approximately 60% of the patients with this mutation have autism. So this is a huge target. Importantly, what we have targeted in those mice, they are deficient in both domains, social domain, and they really show rigid behavioral patterns as well. Both those are the characteristic symptoms of autism, but we have targeted the rigid behavioral patterns. This was our target. Why? Because we knew very well the molecular and neural mechanism that is underlying this problem. Our target in these mice was the protein called matrix metalloproteinase 9, which is regulated by the FMRP protein, which is the product of FMR1 gene that is impaired in those mice. We also knew where in the brain to look for to target a selected symptom that is behavioral flexibility in these mice. So the neural circuit that we chose was the central nucleus of amygdala, deep in the brain. This time we have coronal section, meaning that we cut the brain like this, not like this. So we targeted this area and the specific subnucleus of this area because we knew that it's highly involved in producing particular type of behavioral flexibility in reward-motivated behaviors. And our intervention was based on injecting into the brain nanoparticles. So those are small polymeric particles that are slowly releasing what is loaded into them. And in this case, it was a TIMP1 protein that is directly regulating the MMP9, which is hyperactive in those animals. So they have too much of the MMP9 protein, and we targeted it with TIMP1, which we know brings the level of MMP9 down only within this particular brain circuit to target the normalization of the behavior. And indeed, what we achieved was multifaceted. First and foremost, we show that the level of the protein, which we knew was the upregulated protein, which we knew was the cause of improper synaptic plasticity, was normalized. What we can see, and we'll see in all the subsequent slides, is that the blue bar is for the wild type animals. The empty orange bar is for the animals that were untreated or treated with vehicle, meaning empty nanoparticles. And the orange one is for the animals that were treated with our targeted approach. So as you can see, for the animals that are wild types and the treated ones, the level of MMP9 in the central amygdala is exactly the same. The second thing that we have shown that I'll just briefly describe to you is that the ultrastructure, meaning what is happening at the level of electron microscopy, very tiny structures within the neural cells and between them, has been regulated. The number of multi-innervated spines, so the spines, the dendritic spines that are at the end of the dendrites, has been normalized with our treatment. And finally, the neuronal plasticity measured on a physiological level, so how ready the neurons were to change their activity, was also normalized after our treatment. So this shows that it works on many different levels, from the protein to the physiology of the brain. But most importantly, it regulates behavior in a way that we intended it to. So what you can see here is the test in which mouse should learn flexibly that the reward is available to it in one, but not the other part of the experimental corner of the cage in which it lives. The mice that are wild types, again, marked here in blue, learn very fast. Here you have subsequent days of the experiment. Here you have the percentage of the correct responses. But the animals that are the autistic model, the model of the autistic phenotype, cannot learn this flexibly at all. However, after our treatment, they learn very fast, to some degree, maybe even slightly faster than the wild type animals. At the same time, when we are testing the symptom that we have not targeted, which is social behavior in those mice, in this different tests in which they can either be interested in social stimuli or not, the behavior of autistic model is not changed by our treatment. As you can see, the wild type animals like social exploration quite a bit. The FMR1 knockout animals, so the autistic model, not so much. And the treatment doesn't do anything for them in that regard. The answer to the question why, it's because the neural circuit that was impaired and we targeted was responsible for behavioral flexibility in reward-motivated learning, but not for social behavior. So this is actually the proof that the proposed approach is symptom-specific, which is the very key of the targeted approaches in therapy. So to bring it all together, I have 3 quick thoughts for you to maybe remember after this lecture today. First is that we are at the point in neuroscience when we have neuroengineering tools that are allowing us to pinpoint particular biological deficits that are causing neurodevelopmental disorders in proper learning. And they can be, those impairments, they can be common to many different neurodevelopmental and neuropsychiatric disorders. So learning how to fix them can be beneficial to many patients across multiple different types of disorders. Secondly, we have to understand with the toolbox of today's neuroscience, we see that distinct neural circuits are responsible for different elements of the same neuropsychiatric disorder. So like I have just shown you on this last slide, you can regulate the particular genetic impairment in one part of the brain, and it's not going to happen to alleviate all the symptoms that are appearing in a given patient or a given experimental animal, like in the case of our experiments. But it is going to allow you to regulate what the functional neural circuit is responsible for. And finally, the latest trend in developing new methods, new approaches to understanding and treating deficits of neuroplasticity is the targeted approach. So tailor-made therapies that are going to be adjusted for the particular patient and what is really wrong and inappropriate with the neural function within their central nervous system. With that, I would like to thank you very much for your attention, and I understand that maybe we have time for some questions.
Hello, Alicia. That was absolutely fascinating. We have had a lot of wow, a lot of wow.
Oh, yes.
which is incredible, like really, really interesting especially for behavior analysts, because having this perspective from the neuroscience point of view, we think about it a lot, but we really don't know a lot about it. And it was just so interesting to hear from you. We have indeed a couple of questions, if you don't mind. I want to kick off. I want to kick off with a comment that you made that I thought was quite interesting, because as behavior analysts, we consider behavior to be the interaction of the organism with their environment. And in this context, what do you mean when you say the activity of the brain produces the behavior? You said that earlier on a couple of times, and I'm sort of thinking, well, we wouldn't think that the brain produces the behavior, but that it's part of the behavior. Certainly when it comes to from a purely behavior analytic point of view. But you thought it has a slightly different function. Can you maybe elaborate?
Thank you for this question. This is such a basic question for the understand of sequence of events, right? Indeed, saying that the brain produces the behavior doesn't take away the fact that the behavior occurs in the interaction with the environment. But what is between the environment and the behavior that is visible is the brain. So the organism sees the environment, and the response to the environment is twofold. First part of it is registering what is the context, and this is registered by the brain. And then the brain produces the response, and then only then the behavior comes out, because the brain function precedes, and there is multiple experiments that are showing that, including the ones that I have just shown you. It's the brain activity that precedes the behavior that is coming out into the open. And the same is true about emotions, physiological responses, motor behavior, you name it. So the environment sends the signals from our sensors, our eyes, our ears, our sensory organs go back to the brain. The brain processes this information, generates the sequences that are underlying behaviors, and only then the behaviors occur in response. Doesn't make sense?
It makes an awful lot of sense, because we just talk about the organism as if it's not a thing. It's a whole mixture of anything to do with the organism, and the brain function that you've described is obviously part of that organism. So the interaction between the organism in all its permeations and all the bits and pieces, and you've just gone into the minutiae, and I'm not surprised that a lot of behavior analysts thought this was a very, very interesting talk, because the minutiae that you are analyzing is really, we look at the behavioral side of that in the same sort of microscopic sense. So, yeah, it makes a lot of sense. We have actually an interesting question here from Brittany. She's interested to learn more about the ethics of optogenetics and chemogenetics. How do we decide whether or not it's okay to directly manipulate the behavior of animals and potentially humans? What about the ethics?
That's a fantastic question.
Yeah.
And thank you. I didn't catch who asked the question.
Brittany asked the question. Yeah.
Brittany, thank you so much for this question. I think it's at the very core of our understanding and our acting upon the newest developments in all sciences, not only neuroscience, for the good of the people. Every kind of life science has this question at its very basis when it comes to its applicability. I think the same question can be asked about behavior analysis, really. Should we do it? Can we do it? And what are the consequences of it? Behavior analysis has very powerful methods in its toolbox. They are not as precise as what I have shown you, but I don't think there is a qualitative difference. I think it's just more precise, more robust, and more explicit. But the goal and the expression of the manipulation is something very, very similar, in my opinion. I don't have a good answer to this question. I don't think any of us does, because as it has been multiple times in the history of science, what can be used for the good, and obviously this is all done to regulate the malfunction, the problems with behavior, it can be used for the worse. We know it, and it surely is going to happen, like with any other development of science so far. I have some examples on my mind, but I'm sure that you can come up yourselves with the examples when the science really didn't serve the people in a right way. I think that this is the question about ethics, really, at its very core. And I am sure that there is going to be scientists that are going to be unethical about it. There is no shadow of a doubt in my mind. But the question is, should we stop understanding the reality of what is the world in the name of not potentially causing harm? I believe in the light and dark, and I believe we on the light side should do the most as we can to stay in the front of what can be understood, because you can be sure that the dark side is pedaling in that direction and pedaling very hard.
Yeah, I think you're absolutely right. I mean, it's an ethical question for science, because the better we understand the world, the more also that understanding can be abused. And I keep saying the idea of gravity. You know, you can use gravity to hang a mobile over a baby's bed, or you can use gravity to throw the atom bomb over Hiroshima. That's the ethics of science. Shall we stop, as you say, shall we stop doing the research because somebody might abuse it, or shall we continue? And because hopefully the good people are going to prevail and use the knowledge for the good. Maddie is asking, Maddie, and I'm not quite sure if I understand the question, but I put the question to you and see if you maybe understand it better. Maddie wants to know, are there any trends in action potential required for different animals, different brain sizes?
Can you say that again? Any kind?
Are there any trends in action potential required for different animals and different brain sizes?
An intriguing question. So actually the action potentials, meaning the activation of the neuron, when the neuron is sending a signal, we call it an action potential. They are very similar in many different species. So even though the brain is very intricate, the very basic nature of how neurons are communicate with one another is 0 and 1. It's binary. Either it goes or it doesn't. So the action potential, usually its shape, the shape of the line that marks the type of the signal that it produces, and its very nature is going to be similar in many kinds of neurons. It can be a little smaller. We have different patterns of how the cells are activated. But for example, some kinds of inhibitory cells, most notoriously parvalbumin expressing cells, they are so-called fast spiking. So they are sending action potentials very fast after one another. But the action potentials are very similar in those cells that in any other kind of cell in the brain. And I believe that the question was about different species. In fact, in different mammalian species, the action potential. So look at how the basic element of the function of the nervous system, the action potential is, that it's so similar across different spectra, different types of neurons, different kind of animals. Fantastic question.
Okay, thank you so much. I think that was quite a technical question that I'm glad you understood it better than I do, but I'm learning. Teresa is asking, she thought it was, first of all, making a comment, she thought it was super interesting, but she's wondering, what kind of feedback have you got from the neurodivergent community about this research? Has there been any ethical concerns about where this might go? Obviously, we have talked about the ethics already, but this is a question around how does the neurodivergent community respond to this kind of work?
Yeah. And as always, there are different responses depending on the particular situation and depending on how people feel about science itself. So there is a lot of people who are very enthusiastic, and they are actually writing us, asking about what are the next steps of this research? How can it be accelerated for the clinical trials to happen as soon as feasible? And there is also people who are quite concerned. And usually those are the people that are very skeptical about science in general and are trying to, I wouldn't say solve the problem, but reach for so-called natural methods, unstructured, unbound, and oftentimes having very little to do with what we understand as scientific approach, such as it is understood in behavior analysis. So yes, there is definitely skepticism, and usually it has to do with being quite hesitant and distrustful about the scientific community at large.
I think that's definitely a really valid point in this regard. I think also that a lot of people that would be from the neurodivergent community would consider themselves neurodivergent. They would have issues with terms such as malfunctioning or disease and those kinds of terminologies, because they don't see autism as falling into those terminologies and it not being suitable. But I think that's mostly because their autism is still allowing them to be very productive and being part of independent. Yes. That's really the quality of life. And they might see that they actually have some special abilities and so forth as part of their diagnosis. Whereas what you're looking at is really the other end of the spectrum, in a sense, where people really have functional problems that cannot be described any other way than that these are issues that need to be addressed for their own sake. Yes. They might wish to have them addressed themselves, or they might not even be able to express themselves well enough until some support has been provided and some skills have been developed. So I think it depends on what we're talking about.
I think you've touched on a very, very important element, and it has to do with the brain as well. There is certainly a number of people who have, even though that they have some imperfections in how their neural background works. We all do, by the way. And their brain can function just fine because the neuronal plasticity and the compensation effects within their brains, they deal with the reality just fine. So they function differently than other people, but they are able to interact with the environment to their satisfaction. They can live their lives the way they want it to be lived, even though, again, they can be divergent from what most people how most people function. And this is fantastic and beautiful that we have all those compensations in the brain that allow that. But indeed, we are talking about the very different situation here. We are talking about the malfunctions, because you cannot call them any other way, that are causing people not to be able to even interact with the environment in a way that they could if this malfunction or the biological problem was removed in the first place. So I agree. Diversity on the macro level is beautiful as long as it doesn't hurt you, your life, your needs, and your family and the people you love.
I think that's a very sensible response and answer to that question. Abigail has a couple of questions here, and I think we're nearly coming to the end, but I think people could probably talk to you for a very, very long time. I know I could. Abigail thanks you for the lecture as well, first of all, and she's wondering if these methods have any level of effectiveness when manipulating behavior after traumatic brain injury or for geriatric conditions like dementia, what probably she's wondering about.
Yes, another fantastic question. Indeed, all across the board, from schizophrenia through Alzheimer's disease, Parkinson's disease, all of those are trying to be addressed with the brain manipulation methods and brain understanding methods that we have just talked about. In all of those areas, and especially the disorders that are most prevalent in elderly individuals that you are talking about, there is a huge amount of research being done, not only in terms of the targeted therapies, but also in terms of application of the newest toolbox of neuroscience to understanding them and to trying to functionally improve the lives of the individuals who, for example, have Alzheimer's. There is miles to go. This research is not going to be done overnight, but this is really accelerating. So yes, traumatic brain injury as well. I mean, you name it. You would be hard-pressed to find any kind of dysfunction in the central nervous system that is not being worked on. But the disorders are most prevalent in the elderly individuals are really, I would say, they are very, 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 a lot of money is going into that research, and it's very popular these days. So it's relatively easy to get a grant for it. So there's going to be a lot of researchers interested in that too.
Oh, very good. Two more questions, and then we'll have to let you go because it's getting late. Regarding, Maddie would like to know regarding ASD and neurological plasticity. Do exercises that work to increase psychological flexibility, such as acceptance and commitment therapy, help combat those effects?
This is the intriguing question from the point of view of how the brain works, right? There is nothing better, nothing more effective has been developed so far than behavior analysis when it comes to battling any kind of ineffective behaviors or behaviors that need shaping or don't look in the first place like the individuals that are expressing them would need them to look like. However, when you are dealing with the kind of malfunction in a brain that makes it much harder for you to learn, eliminating this problem in the first place could accelerate how the behavior is being shaped with the use of the traditional behavior analysis tools. So if we would be able to pinpoint the biological problem in the first place and thus accelerate the process of learning after the problem has been identified, wouldn't that be just great? Wouldn't that be just great? It would make the implementation of the standard, well-researched and well-used tools within behavior analysis to such a more efficient degree. Don't you think?
I absolutely agree. I think if neuroscience and behavior analysis working together, and you're sort of personifying this combination so beautifully, I think that would be really, really helpful. One last question from Brittany, and I think she's already had a question, but we'll have her anyway because these are good questions. She was wondering if you foresee potential applications for mental health diagnosis more generally. Is it a matter of finding out where and what to target within the brain structure? Is that, and I suppose, and then working with the behavioral interventions in cooperation?
You pinpointed this, Brittany. It's exactly about that. It's about understanding when, where, and how in the brain those things are happening. And the answer, I think it's very, very probable that in the future, this is how the diagnostics will look like. You will try to understand, even having two patients having exact same symptoms, which is very improbable. But let's imagine this thought experiment. We have two people with exactly same symptoms. They may have very different biological problems that are causing those symptoms. So the first step would be understand what is not right with the function of their nervous system and fixing that in the first place, because before you go and plan the therapeutic process for those individuals, which as a result can be in fact very, very different. Mm-hmm. And what is, I think, very probable as well is that we will use advanced imaging methods, not only the types of genetic analysis that I have shown you, but also brain observation during behavior methods that are going to be very helpful in understanding which neural circuits, or at least which areas, we should be looking at with our interventions. So I think that's a very poignant question. And this is exactly where the medicine is going to go, in my opinion.
Yeah, and I think that's Alicia. That's all of our questions, and I'm just thinking. I think we'll have to have you back for more. There are people asking, where can we get more? Can we? But I think this has been a whistle tour of your expertise, and thank you so much for sharing it with us this evening. And we're very much appreciative.
It was a great pleasure.
Thank
you for having me.
It was just lovely, and I just thought when I met you in Poland only a few weeks ago, I thought, no, we have to have Alicia for the centre to give us a webinar because I was just blown away by your work, and I'm glad to see that the audience seems to have a very similar feeling. So thank you so much, and I hope we'll see you again very, very soon. And take good care of yourself.
Thank you very much for sticking till the very end. It was a pleasure, and please reach out to me if you have any more questions that come to mind later on.
Yeah, definitely people want to hear more from you. Okay, bye-bye now. Bye. Thank you so much.
Bye now.
Bye.